Good morning. We're going to split the PowerPoint for topic 7, the skeletal muscular system focusing on muscles, into two parts because it's so long. This is going to be part 1, and Pansy Answer Dog is all ready to help out. She's got a lot of, she likes mussels actually, she likes to eat them. Okay, so here we go. So muscle tissue makes up between 40 and 45% of the body's mass. And for the most part, there may be some generation occasionally, but for the most part, cell division is complete at birth. This is potentially problematic because it means that if you damage muscles and you actually destroy the cells, you don't get new cells to replace them. Also, it means that the number of muscle cells you have when you're born is basically the same number you have when you die of old age and arthritis. And so some people would say, well, my goodness, person gets a lot bigger. They do. The cells themselves get larger. It isn't that there are more cells. It's just the cells that exist become larger. Muscles contract, and when they contract, typically they shorten, at least if they can overcome any resistance that they face. And so muscle contractions are going to have four main functions. And one of the functions that muscle contractions serve is to create pressure gradients. We will be talking about this when we get to the cardiovascular system. Pressure gradients, and also the respiratory system and the gastrointestinal tract. Pressure gradients are used to move things within the body. So it could be blood or air or material in the GI tract. Muscle contractions also serve in thermoregulation. And when muscles contract, more than half of the energy they release comes off as heat. So if you're cold, you can shiver, and the shivering will warm you up. Muscle contractions support us against gravity. So most people would say, but the bones support us against gravity. That is true. The bones do most of the work in supporting us against gravity, but muscle contractions are required to keep the bones in the appropriate position so that they can support us. And then in movement, muscle contractions allow for movement in conjunction with the bones and lever systems, and we'll talk more about each of these in turn. So there are going to be three kinds of muscles. There's striated or voluntary muscles, which are called skeletal muscles. On an exam, if you had fill in the blank, you wouldn't want to put down just skeletal, you'd want to put down skeletal muscle. We have cardiac muscle tissue and we have smooth muscle tissue. And smooth muscle tissue is sometimes referred to as involuntary muscle. This is because we don't have the ability to consciously make them contract. There are some areas where we have transition zones, where we transition from one kind of muscle to another. There are a bunch of them in the body. One would be going from cardiac muscle tissue of the heart into the aorta. The very beginning of the aorta is more like cardiac tissue than it is like smooth muscle tissue, at least the tunica media, the muscular layer of the wall. Same thing happens, transition zone from the pharynx down toward the esophagus. So the superior portion of the esophagus is more like skeletal muscle that we find in the pharyngeal region, but then it transitions. By the time you get about a third of the way down the esophagus, it goes over to smooth muscle. And then at the other end of the gastrointestinal tract, back at the rectum and the anal canal, we have transition back from smooth muscle to skeletal muscle. Skeletal muscle cells are sometimes called muscle fibers, and they are striated. We're going to be seeing some pictures of the striations. The striations simply mean that there are stripes if you were to look at them with a microscope. They're mostly nucleated. This is a fairly unusual situation. Most cells in the body have a single nucleus. There are some others that are multinucleated. For example, the polynucleated neutrophils can have more than one nucleus, but mostly cells just have one. So you might wonder, why would we benefit from having multiple nuclei? And Pansy knows the answer. Right, because they're big. Muscle cells can be up to a foot long. And remember that within the cell, we rely on diffusion to get materials from one place to another. If you have a cell that's a foot long and you're waiting for something to diffuse over that entire length, it would take way too long to be effective. So by having many nuclei, the nuclei can serve their respective areas within the cell. The cells are also cylindrical in shape, and they're attached to bone. So here we have a photo micrograph and a drawing of the cells, and you can see the stripes. These little blue things are the nuclei, and you can see there's like a dark stripe here, and then a light stripe, and a dark stripe, and a light stripe. Okay, that's the striation that we talked about. The cells are voluntary, and there is an exception. That would be with reflexes. Reflexes are not voluntary, but they're always controlled by nerves. So if you were to cut the nerves to a skeletal muscle, or an organ that's primarily skeletal muscle, let's say the biceps brachii, it won't contract. That's it. It's the contraction phase will not happen because the nerves are required. There's no self-excitation. We'll see that there is self-excitation in the other muscle types. The striations, they're going to be alternating dark and light bands, and the reason this occurs, or the reason this is obvious, is because there are proteins within the cell that are either thick or thin, and they are arranged in a consistent way, so with a consistent degree of overlap, so that we'll see how those striations actually occur. Cardiac muscle cells, sometimes called cardiac muscle fibers, they're going to be found in the wall of the heart. That shouldn't be too much of a surprise. And they typically have either one or two nuclei. They are striated, so the striations is consistent with skeletal muscle. The fact that they don't have as many nuclei is not. Their cells are cylindrical. You'll find this is kind of interesting. If you read enough books, physiology books, you'll find that sometimes cardiac muscle is lumped into with smooth muscle, but it does have characteristics of smooth muscle, and it does have characteristics of skeletal muscle, and it is fairly unusual. So I think it really merits its own category. Okay. Cells are going to have intercalated discs, and these are associations between the cells that allow them to interact. There are both gap and adhesion junctions between the cells, and because of these special associations, the cells, or all the cells can act as a syncytium. Syncytium simply means that they all do the same thing at the same time. And this coordination is extremely important because it increases the efficiency of cardiac activity. The cells have spontaneous action potentials. This is unusual. This is not consistent with the skeletal muscle. Remember, the skeletal muscles have to have nerves. Cardiac muscle cells can actually contract even if the nerves are severed. And basically what happens is the cells spontaneously depolarize, probably because of reduced potassium permeability. The cardiac cells can also be controlled by hormones, so things like norepinephrine and epinephrine from the kidneys can increase the cardiac activity. One of the interesting things about cardiac muscle cells is that if we were to look at their action potentials, there's a long plateau where they remain depolarized, and then they have repolarization. That long plateau means that the cells can stay contracted, but they also have a really long refractory period, and that forces them actually to relax between contractions. Remember, well, actually don't remember because we haven't gotten there yet, but we'll be talking about skeletal muscles going into tetany. If cardiac muscle cells went into tetany, you would die. So having them relax in between contractions is going to be important in allowing the cells to recover from the activity that they've been sustaining, but also it's going to allow the heart to fill with blood, and we'll talk more about that in the cardiovascular system. As I mentioned, it doesn't require nerve stimulation. But they are involuntary. So you can't say, gee, I think I'll contract my left ventricle for 5 seconds and keep it contracted. That's not possible. However, nerves do affect both the rate and the strength of the contractions, and the primary nerve influencing the activity of the heart is the vagus, which is a parasympathetic nerve. So parasympathetic nerves do what to cardiac activity pains? Right. Decreases cardiac activity. In fact, strong vagal stimulation can cause the heart to stop. Smooth muscle cells, or muscle fibers. These usually are in the walls of tubular organs, including blood vessels, but they're also at the base of hair. The individual hairs on the surface of your body, and we'll talk about that and how they work. Now, in contrast with cardiac and with skeletal muscle, these cells tend to be relatively small. Rather than being cylindrical, they're sort of tapered. You can see that in this drawing here. A little more difficult to see the tapering here, but the drawing, you can see it pretty clearly, I think. And the cells are going to have a single nucleus. Remember, in the cardiac, sometimes they had 2, and in skeletal muscles they had multiple nuclei. Because these cells are small, and they are cylinders, but they're tapered, sort of like a teardrop at both ends, I guess. They do only have 1 nucleus. There are 2 kinds of smooth muscle. There are multi-unit cells, and there are single-unit cells, or visceral cells. And we'll talk about both of these types. We'll start with the multi-unit. So multi-unit means that there are multiple units. They act independently of one another. And these are going to be found in the walls of blood vessels, the tunica media, which we'll be talking about. The muscles that erect the hair on the surface of your body are multi-unit, and also within the reproductive system we find multi-unit cells. Normally these cells respond to nerve stimulation, but they don't have a normal action potential. Smooth muscles are a little odd in the way their activity goes. Single unit or visceral smooth muscle work in conjunction with other, and they interact. They're closely aligned with one another. This is the major type of smooth muscle, and they can have spontaneous activity. And so, because they're closely aligned and interacting with each other, if you get one of these muscle cells to be excited, then the adjacent cells will also become excited and contract. And this is a way of spreading a contraction over an organ such as the small intestine. Stretching also causes excitation, and that is really kind of important because these muscle cells are in tubular organs. If the tube expands too much, it stretches too much, it can rupture. And this is a sort of a self-preservation, if you will, or a protective mechanism to help reduce the risk that the cells will overstretch and rupture. Okay. And that's really important. Without that, people would have lots and lots of problems. It is possible that when the cell stretches, the membrane leaks, and that's why it has an action potential. That's really just a hypothesis and not any kind of factual thing. Smooth muscles, the single unit cells also have a long plateau. Remember, I talked about that with cardiac muscle tissue. And this allows for the contraction to continue and not lead to immediate relaxation. This is going to be important in the uterus, so that when a woman is about to deliver a child, the uterine contractions begin and then stay contracted, and that forces the infant to be born. So the heart is, I mentioned they have long plateaus, cardiac muscle cells, and so there is that similarity there. The single unit are responsible for peristaltic contraction. So in a peristaltic contraction, the contraction moves like a wave from one end of an organ to another. It happens in the esophagus, the stomach, and the intestinal mass. Cells are usually parallel. There are no striations. They form sheets, and their muscle contractions are slower, but those cells are not easily fatigued. So what do? They create pressure gradients, and pressure gradients, we mentioned, are going to be used to move fluids or gases through the body. They are sometimes involved in thermoregulation indirectly, and we're going to take a look at a drawing that shows how that can be. So here we have some hair on the surface of an individual, right? And you notice that the hair is being pulled erect. This would be by way of smooth muscles. And you'll also notice that when I drew these hairs in, I left a space between each hair and the next one. But in reality, these hairs would be overlapping like shingles. And when they do that, they create a dead space of air between the surface of the hair and the skin. And that acts as an insulator. So obviously, if it's an insulator, that one of the conditions that it would probably be involved in would be right, thermoregulation. But the other time it happens is when right, when you're frightened. She was frightened this morning. She thought there was a monster outside, and all the hair on her back went up. Okay, so I'm going to show you a picture of a cat. Oh, Pansy's getting excited. And we're going to see if this cat is cold or frightened. Pansy, you can give me your take on this. Cold or frightened? Right, frightened. So this is strong sympathetic stimulation. You see the hair is standing up on its end on the vertebral column. Just to review, I think we've talked about this before, but this has really two advantages of having the hair stand up. One advantage is to make the cat look bigger and more imposing, and therefore maybe it would reduce its risk of being attacked by whatever it is that's carrying it. And the other is that it would be a false target. And so if a predator attacked this cat and bit right here, thinking it was going to break the vertebral column and the spinal cord, all it would do would be get a mouthful of fur. Okay. There are coverings, connective tissue coverings for skeletal muscle. One of the coverings is endomysium, and endomysium is going to wrap around cells, fibers, with areolar connective tissue. There's also going to be perimysium. Perimysium is a covering of groups of cells called fascicles. Okay. And then epimysium. Epimysium covers the entire muscle and extends out and becomes the tendon that will attach to the periosteum of a bone or to another muscle. Sometimes that happens. The collagen fibers, we talked about collagen before when we talked about bones and things. Collagen is a protein, strong protein, and the collagen fibers from the epimysium converge and become the tendons, and as I mentioned, they attach to the periosteum of the bone. Tendons do not contract for movement. Now, if you read in books, sometimes you'll read someplace about tendons contracting if they're not under a load for a long period of time. And that is true, they do, they shorten. But if you want to walk down the stairs, you don't wait for your tendons to contract to do that because it would take forever. So on the short term, tendon contraction is really not an issue. But if you take a tendon and you continually unload it, it will shorten. A good example of this, one that really causes problems for people, is their Achilles tendon. And the Achilles tendon is stretched normally when you're walking and things of that nature. And some people decide to wear high heels. So if you wear stiletto high heels, you unload that Achilles tendon, and if you wear really high heels often, those tendons shorten, and then you won't be able to stand on the ground with your heel and the ball of your foot on the ground at the same time. So most people would suggest that wearing stiletto high heels is actually just a device for entrapment of some other person. My recommendation would be that if you're going to entrap somebody with stiletto heels, get it over with and then get them off and wear sneakers or something like that. Okay, so here we have some of the things that we just talked about all incorporated into a drawing. We have the perimycelium up here, okay, endomycelium down here, okay, and then the epimycelium is on the outside of the muscle, remember, going into the tendon. The fascicles are groups of cells all wrapped together, and then the muscle fiber would be an individual cell. Okay, so functions for skeletal muscle. Skeletal muscle performs all four functions. It creates pressure gradients. It is responsible for thermoregulation when you're cold. It helps support us against gravity by keeping the bones in the appropriate places, and it also is responsible for movement. So skeletal muscle is the big factor in all of these different things. It is possible a fifth function could be through protection, as sort of as a bumper. So for example, if you look at the arm, the biceps brachii is superficial to the bone. So if you get hit in the arm, your biceps brachii takes the impact and your bone doesn't get impacted as much. Okay, so we're going to talk a little bit about the skeletal muscle cells and their anatomy. We have a sarcolemma, which is the same thing as plasma membrane. Sarco is usually referring to muscle or attributes part of a muscle cell. We have the sarcoplasm, which is the same thing that we used to call cytoplasm. We have the sarcoplasmic reticulum. And sarcoplasmic reticulum is a modified endoplasmic reticulum. It's extremely important in skeletal muscles because this is where calcium is stored, and it goes around the myofibrils like a sleeve, and with the appropriate stimulation from an action potential, the calcium is released, and this allows the muscle to shorten. Well, and we'll talk about the action and how that occurs. Okay. Also, going through the skeletal muscle cells are these tubules called transverse tubules, and there are two of these per sarcomere. We haven't talked about sarcomeres yet, but we're going to. And they come in contact with the sarcoplasmic reticulum. These are really important features of skeletal muscle cells because they allow the action potential to pass into the center of the cell quickly. So instead of relying on diffusion in these big cells, which would take forever, we can achieve a rapid release of the rapid dispersal of the action potential, which allows the muscle to contract quickly. The myofibrils are going to be bundles of contractile myofilaments. These are what contract, and there are going to be two contractile proteins, myosin and actin. Myosin is a thick protein and actin is a thin one. And then there's going to be a Z line, and the Z line is basically like the wall of a room. It's going to define the end of the sarcomere. Okay. There are going to be mitochondria, and these are going to be dispersed between the myofibrils. And there are lots and lots of mitochondria in skeletal muscle cells because we use those mitochondria for ATP synthesis. And if you're going to have contraction of these muscles, you need ATP for that activity. We also find that in skeletal muscles there is a polysaccharide called glycogen. Now, you may remember, I think we've talked about this, that animal cells don't store very much in the way of carbohydrates for future use. Unlike plants. Plants store most of their excess energy as a polysaccharide called starch in most cases. Animals don't use starch as a storage molecule, but they do have glycogen, which is a polysaccharide. And the glycogen is found in two places. It's found in the liver, and it's found in the skeletal muscles. And this is really kind of handy because in times of great need, the polysaccharides can break down into glucose, and the glucose can then be used to generate ATP. We also have in some skeletal muscle cells myoglobin, and myoglobin is a red pigment. It sounds a little bit like another word. What's the other word, Pans? Right, hemoglobin. Myoglobin is sort of like hemoglobin. It is a red pigment. It bonds with oxygen, as does hemoglobin. And then when the oxygen concentrations get low within the cell, which is when the cell is going to need it, then the oxygen would be released from the myoglobin. Okay. So next we're going to talk about myofibrils and sarcomeres. And the myofibrils are cylindrical. They go the entire length of the cell, and they're made up of many sarcomeres. And so these are sort of like the cars in a train, right? If you have a train, that would be the cell, and then the cars would be making up the train by connecting to each other. These are made up of myosin and actin filaments. Okay, so here is a sarcomere. We have the Z membrane, which you remember I told you is sort of like the wall of a room. We have the actin filament, which is a thin filament. We have the myosin filament, which is a big one. And then we have another Z membrane at the other end. Okay. So the train analogy, oh, there it is, see? The sarcomeres are the individual cars of the train. Okay. So the myosin is going to be the thick protein myofilament, and the actin is going to be a thin filament. Myosin and actin interact almost like a tug of war. We'll talk about how that all comes down. There are also some other proteins associated with these cells, and those are going to include tropomyosin and troponin, which keep the muscle from contracting until it's stimulated, and so they retard the movement normally. Okay, so there's an I band where there's only actin. Then there's a H zone. The H zone is going to be where the myosin exists, but there's no actin associated with it. And then the A band is going to be basically the area where there's overlap between the myosin and the actin. Okay, so the I band, light colored. I is in the word light, so that might be a mnemonic to help you remember that. The A band is dark colored. A is in the word dark, so that might help you. And the darkness is because of the myosin, and the light colors is because of lack of myosin. And then there's one zone right in the middle. That's where there's only myosin with no overlap with any actin filaments, and that's going to be in the middle of the A band. Okay, so the sarcomere is going to extend from the beginning of one actin to the end of the opposite actin. And that's kind of a big deal. Okay, so myofilaments. First we're going to do the thick filaments. Thick filaments are made up of hundreds of myosin molecules, and there are going to be 2 protein strands for each myosin, and the head of the cross bridges are going to be bent, sort of like the analogy I like to use is they look a little bit like a match, but they bend at the end with the head. Okay. So the cross bridges are going to connect the actin and myosin. The cross bridges are part of the myosin, and the head of the match is going to interact with the actin. So there's the actin filament. There are the cross bridges in green, and there is the myosin filament. oops, sorry. So myosin heads are going to slant away from the middle of the sarcomere, and there's going to be a site where the myosin head bonds to the actin when the contraction occurs, and then there's a second site where ATP is broken down to release energy to allow for the movement. Thin filaments. The thin filaments are going to be two strands of actin. They're going to be twisted like a rope around each other. Okay, and the tropomyosin then coils over the top of the actin. Troponin is attached to the tropomyosin. So calcium, when it's released from the sarcoplasmic reticulum, will bond with the troponin, and the filaments then can start to move. And this is referred to as the sliding filament theory of contraction. The actin is pulled over the myosin molecules, much like if you were standing on a dock and you had a boat out in the water and you had a line that was attached to the boat, and then you grab that line and pull the boat toward yourself. That's sort of what it looks like, except when you're pulling a rope, you'd just be letting go of the end that you just pulled in, and the actin, on the other hand, doesn't get completely let go of it, just slides over the myosin. Okay, so here we have the situation before the calcium is added. Remember, their actin filaments are twisted, and then the troponin and tropomyosin are wrapped around those filaments. And then when the calcium comes in, the calcium bonds with the troponin. It exposes the myosin bonding sites, and those cross bridges then can attach to these little yellow spots and start pulling the actin over the myosin molecule. So how does it get activated? It gets activated usually by a motor nerve. Motor nerves throughout the body primarily are coming from the anterior rami of the spinal nerves. And excuse me, the axon terminal of the neuron fits into a depression near the middle of the muscle cell, which is called the synaptic gutter. Now, if you think about this, having it in the middle of the cell is a real advantage. And why would that be, Pants? Right, exactly. If the terminal were at one end, then the impulse or the action potential would spread over the muscle cell from that end to the other. But by having it in the middle of the cell, you get really two advantages. One is that the action potential will spread in both directions, sort of like a ripple on a puddle of water. It'll go toward both ends. And because it only has half as far to go, it's going to take half the time to get to the end. And you're going to have two places in the muscle contracting at the same time. So it increases the contraction strength, and it decreases the amount of time for the action potential to spread. Okay. There's going to be a space between the two cells, the neuron and the muscle cell, and that's called a synaptic cleft. Okay. So the muscle cell's membrane, where this synaptic cleft exists, is going to be highly folded. What would be the advantage of that? Why wouldn't it be better just to have a nice smooth muscle cell membrane? Right, surface area to volume ratio increases the surface area, and that means that there are more places for the transmitter molecules from the nerve to interact with the muscle cell. This is a really cool thing. The neuron always releases enough acetylcholine to initiate an action potential. So what that means is, if a nerve is stimulated to a muscle cell, that muscle cell will also be stimulated. We're going to be talking about nerve tissue in topic 8, and we're going to see there that there can be these initial stimulation that may not cause an action potential, and often requires more than one stimulus to get an action potential on a postsynaptic cell. But in the case of the nerves, it's a 1-to-1 ratio. If you have to the muscles, if you have a nerve with an action potential, the muscle it interacts with will always it will have an action potential. Okay, so the acetylcholine is broken down in that synaptic cleft really rapidly, and it's going to be broken down by a molecule called cholinesterase. So if we look at that molecule's name, cholinesterase, we can see what kind of a molecule it is. What kind of molecules have ASC at the end of their name? Right, enzymes. So why would it be important that the acetylcholine be broken down rapidly? Why don't we just let it sit around? Right, if it were not broken down, it would continue to interact with the muscle cell, and you would have multiple contractions even if you didn't want that. So by having this material released and stimulate the muscle and then be destroyed, you get one muscle contraction per nerve stimulation. You don't get multiple. That's pretty important as well. That means if you want to have multiple contractions, you have to have multiple stimulation of the muscle. And there's only one nerve interacting with each single muscle cell. So if you're going to have multiple contractions of a muscle cell, that same nerve has to stimulate it multiple times. Now there are some drugs, such as nicotine, that affect the muscle cells pretty much the same way as acetylcholine does. And that's a problem. Why is that a problem? Right, we don't have the enzymes to break them down. And so if you have chemicals that you take into your body from outside and you can't break these down and they stimulate your muscles, you start having muscle contractions that are not controlled, and they can easily continue. You can have muscles go into multiple contractions, even tetany. Okay, so since there's only one neuron to a muscle cell, and since all impulses are stimulatory, how could we inhibit a muscle from contracting? Because we do want to do that sometimes. For example, some of the reflexes, where if you get stabbed in the foot or something, you have a cross extensor reflex, and it inhibits flexor muscles from contracting. How can we do that, since the nerves always just stimulate? And the answer would be, right, we inhibit the nerve. Okay, if you have a nerve that's controlling a muscle and you don't want that muscle to contract, then you can prevent that nerve from having an action potential, and then the muscle won't contract. Pansy's having a good day today. She's pretty sharp. She must have been that good breakfast she had. Okay, so a motor nerve impulse is going to activate the cell. And I mentioned the synapse is in the middle of the cell, which means it's going to spread toward both ends. And that means it's going to reach the ends twice as fast than if it would if it started at one end. And it also means you're going to have two places contracting at any given time, which increases the strength of contraction. The impulse is transmitted into the center of the cell by T-tubules. And so that helps also coordinate the beat. That means at any given place on the nerve, on the muscle cell rather, the muscle cell is contracting all the way through. The T-tubules, I didn't mention them. They're sort of, they're pretty cool. You have interstitial fluid inside the T-tubule, and so the action potential spreads not only over the surface of the nerve, but right into the middle by way of these this interstitial fluid that's inside the T-tubules. So, I guess there you go. The T-tubule contains interstitial fluid. I guess I must have been, psychic or something to figure that out. And that means that the cell will contract as a unit, and that increases efficiency. Calcium is going to be released by the sarcoplasmic reticulum as soon as that action potential spreads through. We talked about the calcium interacting with the troponin and allowing the tropomyosin to move so that the bonding sites are exposed and the cross bridges can start pulling on the actin molecules or actin filaments. Okay, so that's going to cause the sarcomere to get shorter. And remember the analogy I used of pulling a boat in that has a line attached to it. This does require, this sarcomere getting shorter, this does require that the force that the muscle cell is generating exceeds the resistance. So if you're holding on to a really large truck that has its emergency brake set and it's in gear and the engine's shut off, and you try to pull it, we won't move it, not because things aren't working properly, but because the resistance is too great. Okay. So the movement's going to stop when the myosin hits the Z membranes, and it's going to hit on both sides, right? And let me back up for just a second. So let me talk a little bit about that. That's sort of like part of an episode in one of the Star Wars movies. So back in the first trilogy, there was one point where Luke Skywalker and Princess Leia and Han Solo, and I think the Wookiee too, and R2-D2, they were all stuck in a trash compactor within some spaceship, and the trash compactors, the walls started moving together to squish all of the garbage, and their solution to how to stop this was to use some braces and put them against the walls. And if the brace hits both walls and the brace is strong enough, then the walls will stop moving. And that's basically what's happening here. The myosin would be like the brace. If you don't have any ATP, there won't be any movement. So you might wonder, when would this ever be? When would an individual not have ATP? And the answer would be when they are right dead. Yep. So some of you have undoubtedly heard of rigor mortis. If a person dies, ATP is no longer being produced in the cells, and the muscle cells basically lock up and don't move. Now eventually, if you have a dead organism sitting around long enough, they all get loosey-goosey again. So why is that? That happens because the proteins within the muscle cells start to deteriorate and get broken down, perhaps either spontaneous decomposition, or it could be sort of sped up by bacteria and things of that nature attacking these proteins. Okay. So what we just went through is called the sliding filament theory. We have the myosin sort of staying put and pulling the actin from both sides over itself. The myosin's doing the work through those cross bridges, and the work is possible because of the ATP that the cell has. So the cross bridges are going to bind with the actin, and then they bend, just sort of like reaching out with your hand, arm, and forearm, grabbing a rope, and then flexing your forearm and pulling the rope toward your body. That's basically what's going on here. Sort of like a tug of war. Okay. Eventually, the bond between the filament breaks. Now, this is also a little bit like a tug of war. If you pull a rope to yourself and you want the movement to continue, at some point you have to let go and then grab the rope further down the rope. So that's basically what these cross bridges do. Cross bridges attach to the actin, they flex, pulling the actin over the myosin, and then they are released, and when they're released, they snap back to their original position, they extend, and then they make another contact with the actin so they can continue to pull. The probability is that the extension of the cross bridges is possible because of energy provided by the ATP. Now, obviously, if you have a tug of war going on and you only have one person pulling on the rope, you're not going to be exerting a lot of force. But if you have 20 people pulling on the rope, then you're going to be able to exert a lot more force on the rope. So as the amount of overlap between the actin and myosin increases, it allows for more cross bridges to attach to the actin. And that means as the muscle cell shortens, it actually is able to generate more and more power. And the more cross bridges that are attached, the more power. So you can see this in real life too. If you want to generate a lot of force, having your muscles all the way stretched out is not the best way to do it. And if you watch a sprinter starting a sprint, they don't stand up with their knees extended and all that sort of stuff. They get down into those blocks and they get their muscles partially contracted, and they're able to generate a stronger force when they begin their running. Obviously, if there's no overlap, there's not going to be any power at all. So you might say, well, when is that going to happen? Normally it wouldn't happen. And the primary reason it wouldn't happen is because of your joints. So your joints do not allow your skeletal muscles to overstretch. And if they did overstretch, then they wouldn't be able to contract. Okay. So what helps prevent the loss of overlap between the muscle cells? Oh, I guess I just said that, right? The joints. Joints prevent that. Now, the impulses on the skeletal muscle cells have a couple of differences from the impulses that would travel over a nerve. One is that they last longer, and the other is that they spread more slowly. And both of these are important because they allow the muscle then to create a force and maintain that force for a little bit of time before they relax. Okay, the innervation. So the innervation, one motor neuron is going to branch at the end of its axon, sort of like a palm tree. So there's going to be a whole bunch of these branches, and each one of those axon terminals is going to interact with a muscle cell. Now, in some cases, the neuron is going to have many branches, and in some cases, they're going to have fewer branches. The ones with fewer branches go to a smaller number of muscle cells, and therefore they're going to be in muscles where there's not a huge amount of strength being exerted. In the cases where they have many cells, you're going to have much more strength. And the cells affected by or are controlled by one neuron, will be called a motor unit. Okay. So each of the branches has an axon terminal, and the axon terminal is going to get close to the sarcolemma, but it doesn't touch. So there still is that synaptic cleft in between. So then the obvious question is, well, if they don't touch each other, how is the action potential going to spread from the nerve to the muscle cell? It gets spread because of the release of the acetylcholine, and that means that the acetylcholine is going to have to diffuse across that space to interact with the muscle cells. So there's a little bit of a delay because of that, because remember, diffusion is random. It's not directed, and because it's random, it takes a little bit of time. So again, that space between them is the synaptic cleft. Okay. Excuse me. The axon terminal, synaptic cleft, and sarcolemma all together form what we refer to as a neuromuscular junction. Okay, and there are vesicles in the axon terminal. Vesicles, remember back from our discussion of cells, are like little balloons full of material, and in this case the axon terminals are going to have acetylcholine inside those vesicles. And we talked about how those could be released into the interstitial fluid. Okay, so we've got that part figured out. Where the heck did this acetylcholine come from? Is this like a miracle? And the answer is, right. She really likes endoplasmic reticulum. The neuron has endoplasmic reticulum, and that produces the acetylcholine. And then the acetylcholine is packaged by the Golgi complex into little vesicles, and those vesicles remain in the cytoplasm of the neuron. Okay, acetylcholine is a neurotransmitter molecule. Okay. So the impulse is going to reach the axon terminal. That's going to cause these little vesicles to move to the cell membrane and burst, and release through exocytosis the acetylcholine into the cleft. Then the acetylcholine is going to diffuse within the cleft. Some of it's going to reach the sarcolemma, and when it does, it bonds with receptors on the muscle cell. So that is going to cause the muscle cell then to depolarize and have an action potential. And remember, one stimulation is always going to have enough acetylcholine to cause the muscle cell to have an action potential and therefore have a contraction. Now, some of you have probably heard of botulism. Botulism produces a toxin, and that toxin prevents the acetylcholine from being released. So if that happens, if the acetylcholine can't be released into the synaptic cleft, it's not going to diffuse to the muscle cell. Muscle cell will not have any contractions. The muscle cell will stay relaxed. Okay, no contractions. And if this happens to a person, basically what the person's going to have is suffocation because they're not going to be able to breathe, because the muscles that cause the exchange of respiratory gases from the outside of the body into the lungs and then eventually back out all require skeletal muscles. If those skeletal muscles aren't working, you stop breathing and you die. Botulism is kind of a nasty problem. Okay. Only one neuromuscular junction per cell, and that's going to be in the middle of the cell. We talked about that. And remember, repetition is your friend, so when I repeat things, it probably seems kind of boring, but it should help you remember these things. When the acetylcholine stimulates that muscle cell, there's going to be an impulse that travels over the muscle cell, okay, and it's going to spread on the surface of the cell, but then it spreads into the middle by way of those T tubules. We talked about that. When the impulse gets into the cell, and as it passes over the cell, it's going to cause the calcium to be released from the sarcoplasmic reticulum. And remember, the calcium then is going to do a couple of things, and here we go. One of the things is it's going to bond to troponin. When that happens, the troponin pulls away from the tropomyosin, and the bonding sites for the cross bridges are exposed. Okay, so tropomyosin, the troponin calcium complex shifts the tropomyosin molecule, and then the myosin bonding sites are exposed, so the cross bridges can attach. Now, excuse me, the ATP bonds with part of the cross bridge head, and it breaks, giving it energy, and then that energy is going to allow the cross bridge to extend back to its original position once it's released. The other bonding site is going to go to the actin, and that pulls the actin over the myosin. Okay, so one head bonds to actin. The ADP and a phosphate group are released from the first site. That's what when ATP breaks down, it releases. It results in the formation of ADP and a phosphate group. Okay, and then the head of the myosin bends, and that pulls the actin over the myosin, and that's the power stroke. That's sort of like pulling that rope toward yourself that is attached at the other end to the boat. And then ATP is going to bond to the myosin head a second time, and that releases the cross bridge from the actin. So this process repeats over and over again. It's like having many arms that can reach out and grab the actin and then go through various portions of the power stroke and then eventually return. Now the good news is, and you can see how this would be sort of catastrophic if this weren't the way it is, all the myosin heads don't release at the same time. If they did, if they all released at the same time, it would be like pulling a boat in with a rope and then just completely letting go of the rope. If you did that, the boat would not continue to go where you wanted. So if these cross bridges all released at the same time, the actin filaments would slide back to their original position. Okay, so as I said, I think a tug of war is a good analogy. You can maybe kind of visualize that. And as the actin is pulled over the myosin, the cell is getting shorter. And remember, I talked to you about the Star Wars analogy with the myosin running into the Z membrane, so that the cell can't get any shorter than that. Okay. Eventually, calcium gets returned to the sarcoplasmic reticulum. This is because of a calcium pump within the membrane of the sarcoplasmic reticulum. And pumps, you'll remember, are active transport, are going to require ATP. And when the calcium is returned, then the troponin is no longer bonding with it. Then the cross bridge, or the bonding sites on the actin, are covered up again, and the contraction stops. Okay. Now, what if you wanted to keep it contracted, the muscle? If you had another action potential right away, that would mean that calcium would continue to be available, and that would protract the duration of the contraction. Okay. So, in terms of cardiac muscle cells, they contract pretty much with the same events that skeletal muscle cells use. But there's a major difference. In the skeletal muscle cells, all the calcium was coming from the sarcoplasmic reticulum. In the cardiac muscle cells, some of the calcium comes from outside the cell in the interstitial fluid, and the calcium is going to diffuse into the cell, and that causes calcium to be released from the sarcoplasmic reticulum within the cell, and that will then interact with the troponin, allowing the filaments to have their bonding sites exposed, and the movement can begin. Smooth muscle cells. Smooth muscle cells are different. They have both thick and thin filaments, and the filaments, instead of being connected to the Z membranes, are actually connected to the cell membrane itself, or to protein molecules within cells, and these protein molecules are referred to as dense bodies. The dense bodies are all throughout the sarcoplasm. Okay, so when the contraction occurs, the fibers are going to shorten in all directions, and this is because they're not arranged in an orderly fashion as they are in the skeletal muscles. They're kind of all at random directions, and also varying amounts of overlap. Okay, and when that happens, the cell then starts to become a little more oval shaped instead of that kind of tapered shape. So the entire muscle shortens all at the same time. Okay, now in the smooth muscle cells, the calcium, all of the calcium comes from outside the cell. There is no sarcoplasmic reticulum. This is going to work for the smooth muscle cells. Why would that be? Right, because they're small. If they're big, huge cells like skeletal muscle cells, it would not work well. Also, another thing about the smooth muscle cells is that their calcium pump, which removes the calcium from the cell, is slower than the skeletal muscle cells. So what will happen if you remove the calcium more slowly than skeletal? Right, longer contractions. Okay. And that's important for smooth muscles because they need to create pressure to move things or things of that nature, and it's helpful if they're going to be able to have these long contractions. Sometimes the cells maintain a constant contraction. It's called tonus, and that's pretty much the case in tubular organs that get stretched frequently. The actin and myosin filaments are not orderly arranged, as I said, with skeletal muscle. There is no troponin, there is no tropomyosin in smooth muscles. So there are also going to be different amounts of overlap between these actin and myosin, and because of that, it means that the cell can stretch quite a bit and still be able to contract, and that works out really well for things like the urinary bladder or the uterus, or tubular structures like that need to have significant amounts of stretching. Chemical events are the same though. Okay, the contractions are slower, and they use less energy. They're not as energy intensive. Oop, I'm ahead of myself again. So not as much ATP is used. Some of the cells spontaneously depolarize, and that turns out to be real useful. So this happens within the gastrointestinal tract. There's a pacemaker in the stomach, there's a pacemaker and the duodenum, and the urinary bladder has some too, spontaneous depolarization. So once these cells reach threshold, they contract, and it's real important in the gastrointestinal tract. Okay, So smooth muscle contractions are slow. And because they're not using up huge amounts of ATP, you can maintain smooth muscle contractions for a fairly long time without fatigue. And that works out really well for things like the uterus. Innervation with the smooth muscles. So if you remember in the skeletal muscles, we had that synaptic gutter, and the axon terminal went into that and interacted with the cell through diffusion. In smooth muscles, we have either contact or diffuse innervation. We'll start with the contact. Okay, so the axons are going to be really close to the cell membrane. There is no gutter, and we use one of two stimulants, neurotransmitters. We can use acetylcholine or norepinephrine. These are antagonistic to each other. And you notice I didn't say which one is stimulatory. It depends. So in some places acetylcholine can be stimulatory. In other places it causes things to kind of shut down. The diffuse type, the neurotransmitters diffuse to the cell, and remember that skeletal muscles only contract if they have nerve innervation. With smooth muscles, they don't need nerve stimulation. They can contract because of hormones. So where does the energy come from? Well, first of all, in all cells there's going to be some ATP, and in muscle cells the ATP that's present within the cell lasts for a few seconds. So then if you want this contraction to last more than a few seconds, you're going to have to get new ATP molecules completed. So they come from three sources: creatine phosphate breakdown, and we'll talk a little bit more about those; cellular respiration. We're not going to go into a lot of detail about cellular respiration. We'll talk about it a little bit. And then the fermentation is the third way that you can generate ATP. Creatine phosphate and fermentation are anaerobic, and that means that they don't require oxygen. And that is a good thing, because that means if you're in a time of distress, you can get these things going for you. But having said that, they don't last forever. So this anaerobic characteristic is sort of unfortunate, but it is what it is. Okay? So the creatine phosphate breakdown, what happens is creatine phosphate has two portions, a creatine portion and a phosphate portion. And when they break into two pieces, ATP is going to be released. Okay, and then if this is going on, the creatine phosphate concentration is going to decrease because it's being used up to release ATP. If the cell is resting, the creatine phosphate can increase by reacting. The creatine reacts with ADP through more mass action, and you get new creatine phosphate. Okay. So here is a really nice diagram. And when I used to teach chemistry, I used to use this physical analogy to sort of help people understand the chemical situation here. If you had two funnels connected with a tube, and you had some fluid inside here, in this case a red fluid, and let's say that one funnel has this level of the fluid much higher than the other, what will happen is, because of gravity, this side that has more than the other will move through the tube to the other side. Okay, this is by the principle of mass action. So whichever side has more stuff, the reaction will go away from that side. So if we had a lot of creatine phosphate and ADP, they interact and you end up with creatine and ATP. If you were to have a lot of this because the cell's been resting for a while, then it could spontaneously go back. Okay, so the creatine phosphate is going to be found in the sarcoplasmin, and remember we had just a few seconds of energy enough from the ATP that's already present, and we get another 8 seconds by using up the creatine phosphate, and then once the cell is resting, you can generate new creatine phosphate in its place. Cellular respiration. Cellular respiration doesn't involve air in your lungs. This is the chemical breakdown, usually of glucose, and most of the ATP that is produced during cellular respiration is going to be produced in mitochondria. Also, you can get glucose from the blood, and so on and so forth. Okay. We're not going to do a lot of detail about this, but basically what happens through aerobic cellular respiration, you end up with about 36 moles of ATP per mole of the glucose being broken down, and that requires oxygen. Okay. So where's this oxygen coming from? Well, you're going to have some oxygen being delivered by the blood, but if a muscle is really active, it may be a problem in getting enough oxygen into it. So the good news is there's a molecule called myoglobin, and the oxygen can bond with that, and then when the oxygen concentrations get low, myoglobin releases its oxygen, so it's like a backup battery or something, keeping you going. When you get complete breakdown using oxygen, the waste products are going to be carbon dioxide and water, and the benefit of breaking it down is going to be that energy is released. Okay, so what happens with the carbon dioxide and water? Right, it goes into the blood. Okay. Some energy is going to be trapped in the ATP molecule, about 40%, when you are breaking down glucose to form ATP. And then the other 60% goes off as heat. This is going to be good news and bad news. So what would be the good news of having energy go off as heat? Keeps you warm. Exactly. What's the bad news? Right, it keeps you warm. So if you're already warm or hot, then having more energy released is not in your best interest. But if you want to, do whatever kind of mechanical activity you want, you're going to have ATP to do that. Okay, and in fact, should mention the mitochondria are really good at forming ATP, and if you didn't have any mitochondria, you would be hard pressed to get enough ATP to be active. Fermentation. Fermentation is also going to cause ATP production. It occurs in the sarcoplasm. It's anaerobic, meaning it doesn't require any oxygen, but it's only useful for short periods of time because the byproduct, the waste product from fermentation, becomes problematic for the cell. So here is a sort of a diagram of how fermentation goes. And you start out with a glucose molecule, and it breaks down into 2 lactic acid molecules, and in the process you get ATP formation. Not a lot, but some. Okay. So if you're undergoing fermentation for very long at all, the lactic acid concentrations build up. Because lactic acid is an acid, the pH lowers, and if you have your pH change in your body fluids, it has a real negative impact on your enzymes and enzymatic reactions, because pH changes often cause the proteins, the enzymes, to denature, to change their shape, and when they change their shape, they don't work anymore. If you have lactic acid buildup for more than 2 or 3 minutes, you'll start to cramp because of low ATP levels and feel fatigue. Okay, so the if you don't have any ATP, and then that means the calcium won't get returned to the sarcoplasmic reticulum. If the calcium doesn't get taken out of the sarcoplasmic reticulum, you get a muscle contraction that can't relax. That would not be good. Oxygen debt. Muscles can function for a while without oxygen, but eventually they're going to need oxygen in order to be efficient. And also going along with the recovery, the creatine phosphate that was broken down to get energy has to be recovered, and you have to get rid of the lactic acid. So the good news is that lactic acid can be saved and for later use. Gets converted back to pyruvic acid, and then that begets breaking down by the mitochondria. Lactic acid can go right into the blood, and if it does, it goes through the blood. Eventually, it'll reach the liver, and in the liver, the cells of the liver can remove the lactic acid and form glycogen. Okay. So, let's say you have a marathon runner. Marathons are very demanding of your resources, and so if a person runs a marathon, they need about 2 days to recover their glycogen levels. Okay. So here's a really cool thing. If you're an athlete and you train, you can actually increase the number of mitochondria in your muscle cells, and if you increase the number of mitochondria, then you can produce more ATP, and the cellular respiration then is going to generate the ATP with involving the mitochondria, and that is going to reduce the amount of fatty acids or the acetic acid that's formed, because fermentation is not going to be used as much. Fatty acids can also be used to break down for organic breakdown to get the ATP formed. Okay, so if athletes are being active, like say running a marathon, and they can depend upon fatty acids to get their energy, that means that the glucose is going to be available for the brain, and the brain is unable to use the fatty acids the way the rest of the body does. If the lactic acid is not being produced as much, then that means it's going to have less effect on the pH, and there'll be less oxygen debt. So using other sources to generate energy, or to generate ATP rather, is good. I should mention that occasionally people talk about making the ATP, and so you might sometimes get the impression that this is being done from scratch. It's not being done from scratch. So the ATP components are already there. ADP molecules and phosphate groups that can just be joined back together. So it's like you're recycling resources all the time. So what do we use the ATP for? A bunch of things. One of the things is to move the cross bridges between the myosin and the actin. We also mentioned that the ATP bonds with myosin, which terminates the contraction. ATP is going to be used to pump calcium back into the sarcoplasmic reticulum. So there are going to be limits to how much you can do during exercise. One is going to be oxygen availability. Obviously that's going to be determined by a bunch of things, including your environment. So if you're at a high altitude, you don't have as much oxygen, your exercise ability will be reduced. But then you have the carrying, the efficiency of oxygen exchange in the lungs, carrying capacity of oxygen in the blood, and then so on and so forth. Another thing that limits exercise is how much glucose there is available. Another is the metabolic rate. Not everybody has the same metabolic rate. When your tissues, your muscles in particular, get up to about 50% of maximal ATP breakdown, then you shift away from aerobic breakdown of the materials, and you shift over to anaerobic glycolysis. Okay, so for single muscle fibers, we have the all or none principle. If the cell reaches threshold, we have it contract. If the cell doesn't reach threshold, then there's no contraction, and that's referred to as subthreshold. And so that means that since you're getting all or none, stronger stimulation doesn't make the contraction stronger for one cell. One single cell is either 100% contracting or it's not at all. And so that's kind of a neat thing. And so then the questions you might ask is, well, okay, if we can only get one cell contracting and it's always got the same amount of force, how can we generate more force? And the answer is that you can get more cells involved, or you can stimulate the single cell more and more quickly, and that will increase the strength. There are going to be three phases of muscle twitch. One is going to be the latent period, contraction period, and relaxation period. Okay. And we'll talk about each of these in turn. We'll start with the latent period. So the latent period is at the beginning of the stimulation, and there is no movement. And the reason there's no movement is because there's sort of some elasticity of the cell itself, and that elasticity has to be removed before the system can start to move. Okay, so you stretch the cells and the elasticity is taken out. And then there's going to be a little bit of a delay because the acetylcholine, remember, has to diffuse to the cell, so it's not instantaneous. Okay, the acetylcholine is going to interact with the sarcolemma, and it's going to cause the depolarization of the cell. The action potential then has to spread into the cell as it travels over the cell, over the membrane. Remember, that's going to be beginning in the center of the cell. And as it moves toward the ends, it goes through the T-tubules to get inside. The action potential is going to cause the calcium to be released from the sarcoplasmic reticulum. And then remember, the calcium is going to interact with the troponin, and the movement can start. Then we have the contraction period. Normally, if the force is exceeding the resistance, the cell will actually get shorter. And cross bridges form between the actin and myosin. Myosin filaments are going to slide over each other. We described that. And the force increases because as they start to slide, you get more and more overlap. Okay. Then the relaxation period. Muscle cells normally stretch back out to their normal length. And that happens as soon as the cross bridges start to break. And the force is going to reduce. Calcium gets pumped back into the sarcoplasmic reticulum. Okay. So, single twitches and relaxation between the twitches. Because it's an all or none situation, then if you get a contraction, every time the contraction will have identical force. Okay, but if you want to increase the force, one way to do it would be to stimulate the cell rapidly, not let the cell completely get back to its original cell length. That means that you're going to have more overlap, and that means with that stimulation, the cell will generate more power. Okay, so if the cell doesn't completely relax, you have more overlap, and you get more force because of that. Okay. So the more rapid the stimulation, the greater the force. This is called summation. And eventually, if you get it so that the muscle is contracting really rapidly, you get a smooth contraction. Part of the reason that the strength of contraction goes up is that not all of the calcium gets back into the sarcoplasmic reticulum. Okay. So another thing that happens when you're exercising and working is your muscle heats up because of the waste heat. Remember, more than half the energy that is released comes off as heat. And if you have more heat, then the enzymes are more efficient, and that also helps increase the strength. And then more overlap means more strength. Eventually, if you keep going faster and faster and faster and faster, eventually the muscle goes into a what's called a tetanic contraction or tetanus. There is no relaxation, and it just smooths right out. Okay. The single muscle fibers rapid stimulation. The tetanus is going to result in this constant contraction force, and we're going to see that in this next drawing. So there is tetany up there, where it goes faster and faster and faster and faster, and then eventually just smooths out. There is the summation of the contraction. Sometimes that's referred to as a staircase phenomenon. And then there's a single contraction right here. It contracts and then relaxes. So there wasn't any continuation of the contraction. It contracts and then completely relaxes. Eventually, if you have a muscle go into tetany, it will fatigue. And at that point, the cell's going to relax, even if it's stimulated. You can't continue to go once the muscle is fatigued. And the things that cause fatigue is that all the ATP gets used up. Could be because of inadequate circulation. Sorry. Or you don't have enough mitochondria to support the amount of effort you want, or both. So lactic acid is going to start to build up. That lowers the pH inside the cell. That inhibits muscle cell function. And eventually the neuron is going to, neurotransmitter is going to run out if you keep stimulating fast, fast, fast. And at that point, then the stimulation stops. Okay, so a little bit more about fatigue. In the body, in normal circumstances, muscles rarely fatigue. And so you might ask yourself, why? Is it because we don't go into tetany? No, actually, we do go into tetany quite frequently. And we're going to talk about how that all works out in the next part 2 of the Camtasia recording for the skeletal muscular system and the muscles. I hope, nice going, Pansy. She was awesome today. Hope you all enjoyed the lecture. Study wicked hard, and start preparing for the quiz 7. That would be a good idea. Bye.